All right, page 250, number 5. Just get the guy going here. Page 250, number 5. Now 5 has part A, B, and part C, so we'll do them one at a time. The question is asking about an organism controlled by one gene with 2 alleles. So if we look at part A, we're talking about blue dominant over yellow. Blue is dominant. So what we can do then is we can assign our alleles. We can say that capital B is the dominant allele, which would result in a blue phenotype, and little b will be the recessive allele that would result in a yellow phenotype. And then the question says to determine the genetic genotypes and phenotypes by crossing a true breeding blue with a true breeding yellow. Now remember what true breeding means. It means that in all the history of that particular organism, it's only been blue, which tells us that a true breeding blue has to be a double homozygous dominant. So that's our one pair. And then a true breeding yellow would mean it's always been yellow, which means it's a homozygous yellow. Okay, that is essentially always the case for true breeding. So if we look at the kinds of alleles that we could get, this is basically just a monohybrid cross. There's only one possible gamete that we could get from this one and only one possible gamete from the other. And so if we cross those gametes in a very simple square, we will get only that option. So the genotype is 100%, and remember, this dominant allele means it will be blue, so 100% blue. Heterozygous blue. In fact, the genotype doesn't even care whether it's blue. It cares about heterozygous, small b, big B. That's the only possible genotype that you could get. The phenotype is what it looks like. That's where the blue comes in. And of course, all of these babies, no matter what, are all going to be looking blue because of the dominant allele that masks the recessive small b. The only way you could get yellow, remember, is with 2 small b's, and there's no way you can get that in this cross. So that's how A works. So that's a simple monohybrid cross. Now in part B, they've changed the rules, and they say that now blue and yellow are incompletely dominant. Incomplete dominance. So what that means is the two traits tend to blend with each other. Now if you go back in your textbook, but when you're doing these questions, you can always go back into the previous section and find the information. Back on page 242, it shows you how to do an incomplete dominant kind of a cross. So here what we do is we don't use a capital B and a small b for our alleles. We choose a letter like color, C for color. So here's what I'll do. I'll choose a C, and we give it a little superscript, like an exponent. And I'll give it a capital B for blue, and the other one will be a Y for yellow. So that's how we represent the alleles when we have incomplete dominance. Okay? And you can watch the video on that too if you're unsure. So now, a true breeding blue would have to look like this. And a true breeding yellow that we cross it with would have to look like this. So let's look at the possible alleles. Well, the only possible alleles are this or this, the gametes that they can produce, which means when we do our cross in our square, we're going to cross those two things. And we're always going to get this combination. Now remember what that means. With incomplete dominance, it means that the two colors will blend. All right? So what you could say for the genotype is 100% heterozygous. All of them. There's no other thing you can get. And then what you can say for the phenotype is that it would be a blended, or a blend of the traits. Okay, 100%. All of them are blended. Now you can guess at what you could blend. If it was red and white, you could say pink. Blue and yellow might blend to make green. So it's possible that these could be green. But if you're not sure, you can just say blended, right? 100% blended, because we have no other possibles, no other possible combinations. Okay, now in part C, they change the rules this time and say that we want to do the same thing, except it's codominance. So we have the very same alleles, CB for blue and CY for yellow. Codominance and incomplete dominance are virtually the same thing on paper. It just results in a slightly different phenotype. So we've already seen that it's the same cross, right? There's true breeding crossed with true breeding, blue crossed with yellow. The gametes are a blue and a yellow. And when we cross them, it's still a very simple Punnett square. And we get still this heterozygous situation. So the genotype is the same. The genotype is all of the babies, 100%, are going to be heterozygous. But the phenotype is where things look different. Instead of a blended trait, remember that codominance tends to get you sort of a little bit of both. If you go back in your book and you read back on page 243, and you read about codominance, you'll see that each allele is fully expressed, and you get a sort of a mottled appearance. So what you get would be a mottled or spotted, a mottled blue and yellow. They wouldn't blend to make green. It wouldn't be a blended trait. It would be a mottled, some parts a little bit blue and some parts a little bit yellow. That's the only difference. So that's the answer to number 5. Okay, so we'll stop there. If you have any more questions about that, then just email me.